Hi, welcome students. I remember in my last video I had discussed the tools and principles involved in biotechnology or genetic engineering. Further ahead, we were required to proceed to discuss various steps to meet the demand and essentials of biotechnology, and these steps were isolation of genetic material, cutting of DNA at a specific location, amplification of gene of interest using PCR, insertion of recombinant DNA into host cell or the organism, obtaining the products of the foreign gene, and downstreaming process. Here in today's video, let us start with some introduction before we discuss the diagram that is shown here. uh, Process of recombinant DNA technology involves the following steps. You must be remembering from my previous video. Isolation of genetic material. The DNA and RNA are the genetic material, but in most of the cases it is DNA, and to cut it by restriction enzyme we need to have DNA in pure form. You know, the location or the place where DNA like molecule is likely to be found inside the cell. It lies inside the nucleus associated with chromosome as far as the prokaryotic cell is concerned. Eukaryotic cell is concerned. So this DNA needs to be obtained in pure form. To cut it by restriction enzyme, we need to have DNA in pure form. So RNA, protein, polysaccharide and lipid like macromolecules should be removed out of DNA. For this purpose, the bacterial or animal or plant cell is treated with lysozyme, cellulase, protease removal of DNA from histone molecule which is associated with DNA in eukaryotic cell, chitinase in case of bacterial cell. The purified DNA is obtained finally by adding chilled ethanol. In class 12 biological practicals, you will have to perform one lab experiment, that how DNA is extracted from the eukaryotic cell, either it is the plant cell or the animal cell. The purified DNA can be seen in the suspension as fine collection of thread. A restriction enzyme application cut DNA, and progress can be assessed by gel electrophoresis. We have discussed previously the experiment on gel electrophoresis. After cutting the foreign DNA and vector DNA using restriction enzyme, ligase enzyme is added, resulting into formation of DNA or hybrid DNA. The hybrid DNA is now passed through PCR, which was the technique polymerase chain reaction involved by Carey Mullis in 1985, and uh, they were awarded Nobel Prize in the year 1993 in the field of chemistry. It is in fact in vitro replication of DNA. PCR can be understood in the following way. Template DNA. Double stranded DNA is subjected to high temperature, resulting into separation of two strands, and each strand is copied by the DNA. Here in this diagram that I have placed here, you can see just the first one segment of DNA double helix. It is the region to be amplified. This is the foreign DNA and it requires its amplification, that is formation of multiple copy of this DNA segment. So the template DNA it is subjected to high heat, nearly 94 to 96 degrees Celsius, and due to this heat application, the DNA segment is separated. The hydrogen bond breaks, and the denaturation of DNA double helix takes place. The two nucleotide primers. Here in the second diagram, you can see the separated strand of DNA 5 3 end and 3 5 end. At both the, here you can see the primer is written here, annealing process is written, and a short oligonucleotide segment is added to each separated template strand of DNA. The primers are oligonucleotide that hybridize to the target DNA for its replication. The process Taq DNA polymerase or Taq polymerase is the new enzyme that is responsible for the uh, polymerization of the DNA segment. And primers are the oligosaccharide that hybridize to the target DNA for its replication. The process by which primer is associated with template DNA is called annealing. This is the process of annealing, in which you see here that the primer strand, the short strand of oligonucleotide is getting attached with the target DNA. Generally, in polymerase chain reaction, Taq polymerase enzyme is used, which is obtained from the bacterium Thermus aquaticus. So, denaturation or annealing of the enzyme, annealing and polymerization are involved in the amplification cycle, forming the large number of copies in a very short time. So we shall discuss the remaining part in the next video. Okay, thank you.